cancer drug that we found has immunomodulatory effects on the immune system response. So what we discovered was that it delays graft rejection, purposely mismatched graft rejection in mouse models, which translates to the idea that we can sort of expand on transplant tolerance in patients who are going to receive maybe a heart or some other kind of a tissue transplant. Right now they have to be a pretty close, almost a perfect match for that to take. Chances are for the rest of your life you're going to be on some sort of immunosystem suppression drug therapy. So with this class of drugs, what we found was that mismatch could be separate, could be further apart, so you could have something that was a little bit more disparate or not quite as evenly matched, and the graft would still stay. And this group of cancer drugs is well tolerated by the patient. So we found some really remarkable effects on that, and we used PCR, which is polymerase chain reaction, to look at cytokine gene expression, so we can tell exactly what kind of response the body is initiating against this graft. And we can tell exactly what the drug is doing by what cells it's blocking expansion of. And this was really cool work because we learned so much more about that kind of technology and how we can, another use for this drug. And working with the faculty is wonderful. I mean, just everybody in the biology department is so great to work with, and the writing of it, the whole point leading up to the thesis defense, and then standing in front of all your peers, and for 40 minutes and presenting all your research was really an incredible experience. And then to have it in a repository like that, too, where you can just find, anybody can go search for your data and find it, and the idea that it might be published down the road, which was more than likely going to happen, with other work that he's doing in the lab. So just to be a part of that was a really neat experience, and I'm thankful to be able to have done that.